Predominance of Wolbachia Pipientis and Massospora Cicadina and Magicicada Cassini capture in the Southwest Ohio area. This research was done by Abigail Grado, Erick Granillo, Roula Ishaq, Marlene Mosqueda, and Arza Nicoll. Sponsored by the Western New Mexico University and Deming Early College High School. Figure 1 shows a Magicicada Cassini, and Figure 2 shows an insect cell infected with Wolbachia Pipientis. Research questions. Question number 1. What percentage of brood X Magicicada Cassini cicadas are infected with Wolbachia Pipientis, a rickettsial bacteria that is changing the sex ratio of the insect population? Figure 3, again, shows an insect cell infected with Wolbachia Pipientis. However, this cell is stained and therefore looks different from the previous one. Question number 2. What percentage of brood X Magicicada Cassini cicadas are infected with Massospora Cicadina, a pathogenic fungus in the class of entomophthalomycetes? Figure 4 shows a cicada infected with stage 2 Massospora Cicadina. And question number 3. Is there any difference in the gender infection rates? Abstract. 71 Magicicada Cassinis were captured from the Southwest Ohio area. Magicicada Cassinis are periodical cicadas and they previously emerged 17 years ago. All specimens were tested for Wolbachia Pipientis and Massospora Cicadina. Identification for Wolbachia Pipientis was based on the 16S gene of rDNA, which is the gene for rRNA in the large segment of the bacterial ribosome. Therefore, the gene is only in Wolbachia and not the insect genome. The gene used for insect DNA identification was the cytochrome C oxidase gene, which is part of the electron transport chain in aerobic respiration. Being Wolbachia goes through anaerobic respiration, the gene is only in the cicada genome. Wolbachia is a rickettsial bacteria that is primarily transmitted vertically, which is from mother to offspring. However, horizontal transmission can occur. Being the Magicicada Cassini X were laid 17 years ago, the infection rate should be consistent with the general insect infection rate from 2004. Massospora Cicadina is a pathogenic fungus in the class of entomophthalomycetes, and it only infects periodical cicadas. Figure 5 shows the periodical cicada, the Magicicada Cassini. Because Magicicada Cassinis fluctuate between 13 and 17 years, these organisms have been understudied due to their infrequent emergence.
In the following map, we see where the cicadas were collected, which is between the states of Indiana, Ohio, and Kentucky, and more specifically in those yellow boxes. In Table 1, we see the exact locations of the capture sites. Magicicada Cassini. Magicicada Cassini are one of the 3 periodical cicadas that fluctuate between a 13 and a 17-year cycle, and the last time they emerged was 2004. They go through an incomplete metamorphosis, egg, nymph, and adult, and as we see in Figure 6, that's a cicada nymph. The adults mate in vegetation where the female oviposits in twigs. After several weeks, the eggs will hatch or will fall to the ground and bury themselves for 13 or 17 years. The nymphs feed on nutrient-poor xylem, and xylem is a vascular system in plants where it's from the roots of the plants to the top. The nymphs build on waterproof tunnels where they will feed on the xylem of the roots of the plants. Ecological benefits. Food sources for many animals increases, such as small mammals, insects, spiders, and reptiles. Periodical emergence of cicada produces an increase of a biomass in an area. Ecological harms. Damage to trees from oviposition, which kills the tissue and allows the plant pathogens to infect the host. As we see in Figure 7, which is a tree damage from a cicada oviposition, which is from female laying eggs. From nymphs feeding on the xylem, which also kills the tissue and allows the plant pathogens to infect the host.
Wolbachia, the reproductive manipulator, and the consequences of Wolbachia infestation. Wolbachia rickettsial alpha proteobacteria endosympies, which can be parasitic and mutualistic. Wolbachia tends to be parasitic, where one organism is harmed while the other one is helped. Wolbachia can also cause cytoplasmic incompatibility, which is the inability to produce offsprings, feminization, parthenogenesis, sterility, and a shorter lifespan among arthropods. Wolbachia modifies the host's reproductive system in order to enhance its own growth. 67% of all insects are infected with Wolbachia, which is a 50% increase since 1997. The infection rate is increasing at approximately 2% each year. In Figure 8, we see a Magicicada Cassini that has been infected with Wolbachia. Wolbachia also helps block many diseases and viruses that are transmitted through insects such as mosquitoes. When an insect is infected with Wolbachia, there is a decline in other infections such as the Zika virus, the dengue virus, the Drosophila C virus, West Nile virus, malaria, yellow fever, and many more. Cytoplasmic incompatibility. There are two types of cytoplasmic incompatibility. In the unidirectional cytoplasmic incompatibility, if the male is infected with Wolbachia and the female is not, then they cannot produce an offspring. However, in the bidirectional cytoplasmic incompatibility, Wolbachia-infected insects can only produce an offspring if they share the same strain of Wolbachia. If the male has a different strain of Wolbachia than the female, then they cannot produce an offspring.
Feminization. In feminization, Wolbachia manipulates the DNA of male insects, arthropods, and nematodes, influencing the cells to produce ovaries and secrete estrogen. Additionally, Wolbachia can completely feminize male arthropods and nematodes into fully functioning females. Subsequently, the manipulation of the organism's reproductive systems is skewing the male-female sex ratio of the populations and decreasing their gene pool. Figure 10 displays the genitalia of Magicicada Cassini. On the left, we have the female genitalia, and on the right, we have the male. Figure 11 displays the male timbal, a sound-producing structure found only on males. This proved quite useful when identifying the sex of our cicadas. Parthenogenesis. Parthenogenesis is the reproduction of an unfertilized ovum which produces only females. Males cannot transmit Wolbachia. In parthenogenesis, females are produced, increasing transmission of Wolbachia. However, the sperm of infected males swim faster than those not infected, giving Wolbachia a distinct advantage. Parthenogenesis has only been naturally found in haplodiploid insects, such as the honeybee, as displayed in Figure 12. In these insects, diploidy is restored by non-disjunction of the chromatids in the mitotic anaphase stage. All offspring resulting from parthenogenesis will be female and infected with Wolbachia. Transmission of Wolbachia Pipientis. Transmission is a transfer or passageway for bacteria and viruses to infect another organism. Wolbachia can be transmitted through two types of transmission, horizontal or vertical. Horizontal transmission occurs through the consumption of an infected organism through either cannibalism or food supply. Vertical transmission occurs between her mother and offspring. Again, males cannot transfer Wolbachia. Figure 13 displays horizontal transmission, and Figure 14 displays vertical.
Massospora Cicadina is a sexually transmitted disease that can only infect the population of periodical cicadas, and it affects both females and males. In stage 1, one of the early stages of the disease, infected cicadas will produce haploincodidia spores that will facilitate the transmission to other adults during the chorus. In the chorus, male cicadas will produce sounds for the purpose of attracting females and therefore mating. In stage 2, infected cicadas will produce diploid resting spores that will fall into the ground and will eventually infect emerging nymphs. In later stage 2 phases, the fungus will be visible as the insect's abdomen has fallen off. Figure 15 illustrates stage 2 of Massospora Cicadina in an infected cicada. Methods for DNA extraction. When testing for Wolbachia, there are a series of procedures that must be followed. For instance, after identification is done, 2 millimeters of the insect's abdomen will be cut off because Wolbachia infects the gonads or sex cells. Afterwards, the lysis buffer will be added because it maintains the pH, it protects DNA from acidic degradation, and it separates DNA from other cellular beads. Then, the water bath is conducted to slow down the process of the enzymes that could break down the DNA. And the centrifuge is used to separate molecules by weight. Consequently, 5 molar sodium chloride is added to separate proteins and carbohydrates from the DNA. And to protect DNA from enzyme degradation, the ice bath must be conducted. The restriction enzyme used for this process is known as tRNase, which helps in the polymerase chain reaction process, or PCR. The PCR duplicates the DNA strands to therefore have enough for the electrophoresis gel. The electrophoresis separates molecules with electricity. Therefore, when the results come out, the 16S rDNA found in Wolbachia can be identified at 438 base pairs, and the cytochrome C oxidase gene found in the insect can be identified at 709 base pairs.
Methods for Massospora Cicadina culturation. First, LB agar was made on a hot plate. Then, the plates were poured and left to solidify. After the LB agar was completely solidified, the cicada's abdomen was swabbed with the Q-tip, and with that same Q-tip, the plates were stripped. Then, the plates were taken to incubate at 30 degrees Celsius for 4 days, and afterwards, the fungus was identified under a microscope. However, the abdomen of the cicadas infected with Massospora Cicadina was particularly harder than those who were not infected. This made it easier to identify the cicadas who were infected and those who were not. Figure 40 shows cicada's 511 abdomen, and as you can see, it has fungus growing inside of it. Figure 41 shows cicada's 511 side of the plate, and it has fungal colonies growing on it.
Massospora Cicadina results. Of the cicadas captured, 46 were females and 25 were males. Of the females, 37% were infected with Massospora Cicadina. However, none of the males were infected. Interestingly, 64% of the males were discovered to have empty abdomens. Figure 42 displays the normal male abdomen, which contains the digestive system and the gonads. Figure 43 displays our empty cicadas, which contains none of the corresponding organs. However, none of the females captured were discovered to have empty abdomens, even when they were identified as infected with Massospora Cicadina. Wolbachia Pipientis results. Of the 10 sites, a total of 71 cicadas were captured, and of the 46 females, 41% were infected with Wolbachia Pipientis, and of the 25 males, only 8% were infected. Figure 44 displays electrophoresis gel, which separates the insect DNA from Wolbachia DNA, and the BP ladder on the right is used as a form of measurement.
Massospora Cicadina discussion. Because Magicicada Cassinis emerge once every 13 or 17 years, there is not enough research done on the insect or the fungus. The cicadas that were captured were all alive and appeared to be healthy. None of them showed any signs of being infected with the fungus. While none of the males appeared to be infected, 37% of the females were. But the general theory of the offspring is that the spores are dormant in the soil until the nymphs are ready to emerge. There was no reference to vertical transmission of the fungus in any published literature. And while none of the females had any empty abdomens, 64% of the males did. This odd discovery was not mentioned in any other published work, which eliminates the question, how did this occur? If vertical transmission of the fungus is possible, could there be a correlation between the empty abdomens and the vertical transmission? It has been shown that insects who reach stage 2 of the infection lose their abdomen but are still viable and functioning for a given amount of time. This could explain why the cicadas appear to be healthy while having empty abdomens. These questions need to be answered to fully understand the propagation and life cycle of Massospora Cicadina. 41% of the females were infected with Wolbachia Pipientis. The Wolbachia Pipientis infection rate appears to be increasing at approximately 2% each year. Therefore, if the rate of the infection was 17% in 1997, it should be approximately 31% in 2004. However, this appeared to be higher in our research. 64% of the males had empty abdomens, making it impossible for the DNA to be extracted from the gonads where Wolbachia is located. This left only 5 males to be tested for Wolbachia, and 2 of those 5 males tested positive. But no conclusion can be made from such a small amount of specimens. Conclusion. Research on Massospora Cicadina must continue, and the question pertaining to the empty abdomens of the males must be answered. Our research on this question will still continue. Our research on Wolbachia Pipientis and Magicicada Cassini revealed a higher infection rate than those published in other works.
These are the main references used for our research. Are there any questions?